Hello, and welcome back to Introduction to Genetics and Evolution. Now, in the past, we've talked about recombination. We've talked about the process of recombination, primarily in the context of transmission genetics and genetic mapping. Now, the question I want to ask you today is what are the evolutionary advantages of sex and recombination? Those two things are intrinsically tied together. Basically, why do we see so much recombination within species? Well, I have a cute subtitle for today's lecture. That is, still more reasons that recombination rocks. Now, sex and recombination are intrinsically tied together, so let's first explore sexual reproduction versus asexual reproduction. Now, there are several forms of asexual reproduction, some of which you may have heard of. Now, asexual reproduction can be through binary fission, where you have basically a single cell. It splits into two cells that are essentially complete organisms in and of themselves. You can have budding, as depicted here on this slide with the hydra. You see the little baby hydras coming off the mom hydra. We obviously don't do that. My arm can't grow off into a kid that grows up. You can have parthenogenesis, which is offspring that come out from unfertilized eggs. That's actually observed in various insect species. All these processes produce exact genetic replicas of the parents. Basically, they produce clones. Sexual reproduction, in contrast, involves the union of genetic material from a mom and a dad, or two parents. It doesn't have to be a mom and a dad. And they're put together, and then ultimately, before that new organism passes on its offspring, its genetic material can be shuffled in its gametes. Now, what process shuffles this genetic material in the gametes? Well, it's our favorite process, recombination. Now, you may not know this, but there are several benefits to asexual reproduction. Well, first, with asexual reproduction, every individual can make babies directly. So, in contrast with sexual reproduction, as we humans do, you actually need to find, if you're a male, you need to find a female in order to produce offspring. With asexual reproduction, anybody can just butt off a baby. So, this is potentially a good thing. Similarly, with asexual reproduction, you don't need to go out and find mates. So this is something that sometimes involves a lot of energy. You have to sometimes have these characteristics that may actually be maladaptive in general, but are good for attracting mates. So this is actually a big advantage to asexual reproduction. In contrast, sexual reproduction requires that you actually go and woo your potential mates. And finally, with asexual reproduction, offspring actually only get, with asexual reproduction, offspring get all your genes. In contrast with sexual reproduction, offspring only get half of your gene. So it seems like overall there's a lot of benefits to asexual reproduction. You can pass on more of your genes. You don't need the intermediary. You don't need to go hunting and wooing mates. Nonetheless, we find that sexual reproduction is very, very common. And this is particularly true in the animal, plant, and fungal kingdoms. We tend to see for many of these species, they only have sexual reproduction. And those few that do have asexual reproduction, it's often not complete. That basically they will go through a few generations of asexual reproduction, and then they will have a sexual phase. So we see all these benefits of asexuality on the one hand, but on the other hand, sexual reproduction is very, very common. Why is this? Well, the benefits come from our friend recombination. So I'm going to give you two major reasons, but there's actually a slew of reasons that contribute to why sexual reproduction and recombination are beneficial. The first one we'll talk about is that recombination makes combinations of alleles across two or more loci that may be advantageous. So imagine that, for example, to have maximal fitness, you need to have the big A allele at one of your loci, and you need to have the big B allele at another locus. How do you put these things together? Recombination facilitates that, and I'll show you that in just a second. Now, this is especially important when you have epistasis, or interactions between loci, that favor particular combinations. So let's use an example. Let's imagine, just as I was saying before, that you have a starting population where everybody's little a, little b, little b. Your optimal genotype has a capital A and a capital B. But mutations from little a to big A are very rare, and mutations from little b to big B are very rare. So you have this population that are mostly little a, little b, and every now and then a mutation crops up. Well, there's a big A. Well, that's good. That's still not the optimal. Well, there's a big B. That's good. That's still not the optimal. There's a big B. There's a big A. If you only have asexual reproduction, this big A and this big B will never come together. Even if they have kids together, well, they're not having kids together if it's asexual reproduction, but if in some way they interacted, you would not put this big A with this big B into one individual. So the question is, how could you put this genotype, big A, little a, big B, little b together? If you have only asexual reproduction, you basically have to wait for that mutation to arise. So here, this individual has a big A, you have to wait for a big B mutation to come up in either this individual or one of its offspring in the future. In this individual here, you'd have to wait for a big A to arise either in this individual or in one of its offspring. You have to wait for mutations. So without recombination, you need to wait a very long time to get the optimal genotype. In contrast, if you have recombination, you can accelerate adaptation, that basically this individual could have a baby with this individual, and some of the offspring would have that optimal genotype. So recombination can make advantageous mutations get together much more rapidly. This allows for more efficient adaptation. Second reason is related. Recombination basically helps you get rid of bad mutations. And in this regard, it helps you create or restore, and you see what I mean in just a second, mutation-free offspring. So let me illustrate this. Let's imagine a case of several independent lineages that are all asexual. So here are your 4 lineages here. There's the first individual, second, third, and fourth. This right here on the far right is depicting the number of detrimental mutations that we have per individual in that generation. So in this case, nobody has any bad mutations. Everybody's got a 0, and the range we see here is a 0. So let's wait 10 generations. Oh, we had a little mutation arise here. So this individual is a little bit less fit than all the rest of these. Well, guess what? Every offspring that comes from this individual is going to have that same bad mutation. He has no way to get rid of it. There's no way for him to eliminate that. So all his offspring are doomed to carry this bad mutation. These ones are still all okay. Well, what's going to happen after more time? Well, we'll have more mutations arising here, 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 here. Eventually, as you see, there's a range of 0 to 1 bad mutations, 0 to 1, a couple more have 1, a couple more have 1. Down here, the range is 1 to 2. We actually have no individuals left in the population after a very long period of time that are lacking bad mutations. Everybody has bad mutations. So in this regard, the population as a whole is getting more sickly. And in fact, the population gets worse or sicker and worse every single generation. So a few generations later, you might see this distribution of number of bad mutations. So again, nobody has 0 bad mutations. The previous example we saw, it was just 1 to 2, but maybe after a while it's a hump around 3. Very few individuals have 1. More have 2, more have 3 bad mutations. You wait a little bit longer, you lose the 0 and 1, and now everybody has 2 or more bad mutations. Essentially, again, the population gets worse and worse because individuals tend to accumulate these mutations, and they have no way to shed them. Right? There's no way to get this good version of this gene and this good version of that gene and put them together. This process is called Muller's ratchet. If any of you are familiar with a ratchet, it's something that actually will only turn things in one direction. In this case, the analogy here is that the population is getting worse. You turn it in one direction. There's no way to move the socket the other way. Now, if you had recombination, you actually can get some offspring that don't have bad mutations. So here are 2 individuals that are both carrying 2 bad mutations. This one has, in this case, the red is indicating a bad mutation rather than a good one. So this one has bad allele at A, a bad allele at B. This one has a bad allele at C, a bad allele at D. Well, we cross them together. Some individuals, some of the offspring from this will actually be worse. They'll have 4 bad mutations. Those will be very bad. Some will be comparable. This one has 2 bad mutations, but it has a different combination of bad mutations than either of the parents. But some will be better. This is the critical element. You can actually recreate the 0 mutation class. You can recreate some individuals that have no bad mutations. This one will reproduce more. This will reproduce some, and this will probably be the most poor reproduction. So eventually, this is the type that will spread within the population. So overall, we'd have to say recombination is good despite the many costs of sex, because recombination can produce these advantageous combinations of alleles, as I showed you, and in that regard, it can accelerate adaptation. Recombination also allows the population to get rid of or unload itself from these bad mutations. Essentially, it stops the ratchet. Now, this will especially be true if you're living in a variable environment. If you're in a very constant environment then maybe not having a recombination isn't so bad. But if you have a variable environment, you're essentially hedging your bets by producing more varied offspring. So this is, one of the many ways that recombination is really good for a species, and this is why we tend to think that recombination is very, very common when we look across species, particularly animals, plants, and fungi. Now, what does recombination do in terms of molecular evolution? Now, we've shown that it can combine good mutations, we've shown that it can get rid of bad mutations, but how does it affect neutral sequences? Sequences that really don't matter. How does it affect how much variation there is present in nucleotide sequence? This variation that has no particular effect on fitness. Well, we'll come back to that in the next set of videos. Thank you.
Hello, and welcome back to Introduction to Genetics and Evolution. In the previous video, we talked about the evolutionary advantages of recombination. In this video, we're going to be looking at something a little bit different. We want to look at how recombination will affect patterns of neutral DNA sequence variation. DNA sequence variation that may not matter in and of itself, but it is actually impacted by, say, the spread of adaptive variants. We'll look at two related concepts, selective sweeps and hitchhiking, in this video. We left off the last video by asking the question, how does recombination affect molecular evolution? And we showed in the previous video that recombination can help combine good mutations, and this is good for the population because you don't have to wait for the two beneficial mutations to arise in the same genetic background. It also helps you get rid of bad mutations from a population. It stops this process that we refer to as Muller's ratchet, which is the steady deterioration of fitness in a population as you accumulate more new mutations and can't get rid of them, such that essentially you lose the mutation-free class and the population as a whole gets worse and worse and worse. But again, the question we want to ask now is, how does recombination affect variation in neutral sequences, sequences that have no direct effect on fitness? Well, the first question we have to ask before we ask that is, how do we quantify molecular variation as a whole? So we have here 9 different sets of sequence, and these are potentially from different individuals. So this could be from individual 1, 2, 3, 4, 5, etc. Well, we look at this, and most bases are the same. So this stretch of 3 T's is the same in all 9 individuals, right? We're assuming at this point that you only have one copy. We're not looking at the 2 copies each individual would have, but let's say we randomly selected 1 copy from each individual. There are 3 sites that are variable. There's a site that's variable here, here, and here. They're depicted in green. Is it sufficient just to say there are 3 variable sites out of however many there are here, maybe about 20 or so? No, because you notice that they're very different. So here at this first site, we have 1 individual that has a G, but everybody else has an A. So that's, an example of a very rare variant at that site. Here, we have multiple individuals that have a T, so a little bit less than half, and several that have an A. And here it's very close to 50-50, where there's 4 T's and 5 C's. So what we need to do is find some way of capturing these relative abundances in our measure of how much variation there is. So one commonly used metric for measuring the amount of molecular variation in DNA sequences when you have sequences from many individuals is referred to as pi. Okay? Well, pi, just very simply put, is the average number of pairwise mismatches among all the sequences you have. This is analogous to the 2PQ idea that we used back when we were originally studying Hardy-Weinberg. It's kind of like the average number of predicted heterozygotes. So let's look at these 3 example sequences I have here. Between the first and second sequences, how many mismatches are there? Well, we can look very simply. There's 1 here, 1 here, 1 here. Made it very easy by making them red, huh? Well, we have 3 mismatches between the first and second sequence. What about between the second and third? Well, those same 3 positions also differ between the second and third sequence. So there we go, we have another 3. What about between the first and third sequence? How many mismatches are there? Well, the only variable sites are the red ones, and here they're the same, here they're the same, and there they're the same. So in fact, between the first and third sequence, there are 0 mismatches. Now, pi is just the average number of pairwise mismatches. So we have, in this case, 3 numbers for mismatches, 3, 3, and 0, and we had 3 pairwise combinations. So we just add these up, and divide by the number of pairwise combinations, which in this case was 3. We have 3 different comparisons. In this case, 3 plus 3 is 6. 6 divided by 3 is 2. Now, it's not very useful just to use this 2 by itself because it could be 2 in 10 bases, 2 in 100 bases, 2 in 1,000 bases. So typically when people talk about pi, they average out by the number of sites being investigated. In this particular case, we looked across a stretch of 20 bases. So pi per site would be 2 divided by the number of bases, which in this case was 20. So in this case, pi would be 0.1, or simply put, 10%. Now, pi is going to be much more affected the more differences you have among sequences. So this is kind of interesting in that if you have a very rare variant, it's not actually going to raise pi all that much within a population. Whereas common variation, if say half the individuals have C and half the individuals have T, you have a lot more opportunities for pairwise mismatches. So pi per site is greater when there are more bases differing among individuals, and pi is not as much affected by very rare variants. Well, let's apply this and see what's happening in terms of studying neutral sequences and the spread of variation. So again, the question is, how do patterns of recombination affect variation in neutral sequences? Now what happens, although these particular sequences, or the variants that are found at these sequences may be neutral, there may be an occasional beneficial mutation that arises. So what happens when you have an occasional beneficial mutation? How does that affect the neutral variation everywhere else? So sequences are not intrinsically neutral. It just happens to be the variation that's present at those sequences that is neutral. So let's assume a case where we have no recombination whatsoever. And we have this variation here at base 1 is neutral. In this case, now we have 3 G's and about 6 A's. At base 2, we again have these A's and T's. At base 3, we have C's and T's. But we're assuming all this variation is neutral. None of this is affecting fitness. Now we have this base 4 here. Now let's say that it's possible for there to be an adaptive mutation at base 4. And let's say here it is. So here this G that has arisen by mutation is adaptive. Well, if there's no recombination across this entire stretch, what's going to happen? Well, since this G here is adaptive, it's going to spread. But since there's no recombination, it's basically glued to this whole set of sequence. So as this G spreads, so will this C, so will this A, so will this A, right? So let's watch what happens over time. The G starts to spread. So G now is across half the population. You notice we've erased all the variation in that half of the population, those other 3 bases. As G spreads completely, we've lost all variation, not just at the adaptive site, but at these 3 sites that previously had neutral variation. Okay, notice this. So we had the spread of an adaptive variant, and we had a loss of variation at these other 3 sites. And not only did we lose variation, but we specifically carried this one variant, the C, the A, and the A from those 3 sites, along with the spreading G. So what we witnessed is called hitchhiking associated with a selective sweep. So a selective sweep is the spread of an advantageous allele through the population by natural selection. This is what we expected from number 4, right? And that 4th base was the adaptive variant. And importantly, the loss of variation associated with the sites near it. So it swept out all the variation that was present there before. Hitchhiking is a related concept. This is the spread of nearby alleles along with the advantageous variant because of linkage or because of this lack of recombination. So in this case, at SNPs 1, 2, and 3, we had the A, and C variants that all hitchhiked along with the spread of that adaptive variant at base 4. So the sweep there is what got rid of all the old variation, and hitchhiking is what happened in terms of these particular 3 variants. Now this was the case when there was no recombination whatsoever. What would happen if we actually said there was lots of recombination among these sites, that they weren't all glued together as one variant? Well, let's try that. So now this is exactly the same slide that we saw before. So what if there was lots of recombination among these sites? Well, we might initially have this variant right here is going to spread, and it is, in the very first generation, it is associated with this A, and C at bases 1, 2, and 3. So, okay, it spreads a little bit. But now there's some opportunity. Let's say, for example, this one has a kid with one of those. There's opportunity for some recombination to happen. So there's a heterozygous individual experiences a crossover before making a gamete. So initially we have this AAC associated with the G, but we have this other haplotype, GTT. And what might happen is we might have a crossover that, just like that, which essentially puts this G and C next to a T and G over there, producing this new gamete. You see this? So this is something that was not found in the parentals, right? This is a recombinant because the G and C were previously together. The T and G were previously together, but they were not. This group was not previously with this group because you see one of them came from this gamete, or I'm sorry, one came from this haplotype, one came from that haplotype. So that gets moved into the egg, and we now have a case where there's the G adaptive variant associated with the T and G over there rather than just with the two A's. Note the C there is still with the G because it was a lot closer. So we might have more spread. So what's happened in this case? This is a little bit different now. We look here at base 1, there's still some variation. We look here at base 2, there's still some variation. Base 3 actually has a little bit of variation, but it lost a lot of it. This one little T slipped in, but mostly you lost a lot of the variation there. So let's break this up. So what happened with recombination? Well, the mutation at site 4 still spread. That's very important. It's not that selection stopped. Selection operated just the same as it did before. Recombination allowed SNPs 1 and 2 to maintain actually most of their variation, that we still have 2 abundant alleles at those 2 SNPs, right? So the other alleles recombine onto the chromosomes, bearing the sweeping allele at site 4, so pi was actually not reduced in that left half of the picture that we just saw. At SNP 3, we had something that was kind of intermediate between the two. At SNP 3, most of the population got one allele. Right? We had that one variant that spread, but there was a rare one that didn't. So most of the chromosomes have the same allele at SNP 3 after the sweep. So there was a loss of variation at SNP 3, but not to 0, it just lost some of it. And there was one particular allele that became very common by hitchhiking. Okay? So in this case, if you go back, let me go back actually a couple of slides. If you go back here, looking at this, there is, if you split this in half, there is a big loss of variation in this side of the picture, right? Whereas there is basically no loss of variation in this half of the picture, okay? So pi is greatly reduced in the right half, unlike the left. And we did actually have a reduction in variation at SNP 3, but we did have hitchhiking still happen as well. So this is pretty cool, and this is the kind of thing that we expect happens as we have the spread of adaptive variants through human populations or through populations of any species. Let me show you one hypothetical example. It's actually not hypothetical, it's a real example. This is looking at variation at a single nucleotide polymorphism near the EPAS1 gene. Now this affects tolerance to low oxygen. So there were some interesting studies looking at high altitude Tibetan populations. And sure enough, what it turns out, these are very high altitude populations. This affects tolerance to low oxygen. There's evidence for a recent sweep and hitchhiking in these Tibetan populations. If you look at a particular SNP, the A variant in Europe is at a frequency of about 0.46, right? Now there was a mutation to tolerance for low oxygen that arose in China. It arose on a chromosome that had this A allele. So in these Tibetan populations, we now see the A SNP frequency is 0.89. So this A SNP hitchhiked along with the adaptive variant at EPAS1. And it's beneficial to have this new variant. So this is something that's often used as a signature of natural selection. Now let me recap 3 principles here. First, adaptive alleles can sweep. That means that they spread through the population and they eliminate some variation nearby them. The second is that SNP or marker alleles, SNPs are markers, that are near the adaptive allele. By near, I mean that they don't recombine away from it. I'm not necessarily referring to physical distance, I'm more referring to recombinational distance. So basically, if they have 0 or low recombination from the adaptive allele, they will tend to hitchhike, and pi will typically be reduced at those SNPs or in those areas. In contrast, SNPs or marker alleles that are far from an adaptive allele, so in other words, that there's a lot of recombination between them and the adaptive allele, they do not hitchhike and pi is not typically reduced. You do not see the evidence of a sweep in those cases. So this is pretty cool, and there's actually exercises to demonstrate this if any of you are interested. This is actually one developed by my laboratory. This is something where you can essentially put this together in your lab. There's a paper on it in the journal Evolution, Education, and Outreach by Heil et al. Heil is a PhD student of mine. Essentially, you start this population with a bunch of white-eyed fruit flies. These white-eyed fruit flies are healthy. They're okay. They're doing fine. You introduce a red eye allele. That's actually the wild type allele. What ends up happening is after several generations, you see that most of the flies end up having red eyes because this red eye allele is advantageous. Now, this bottom part shows you a chromosome. So the eye color gene is on the X chromosome, and here's the red-white eye color gene. Now, you can do PCR, or ways of assaying two particular genetic markers. One of them is called Near, one of them is called Far. Near happens to be right next to the eye color gene. Far is very far away. Now, let me ask you in closing here, what do you expect? Which of these two marker alleles should hitchhike along with the red eye variant? And then two related questions. Do we expect pi to be reduced at the near region? And do we expect pi to be reduced at the far region? So try those out and then check out the next videos. Thank you for your time.